Welcome, everyone. Very happy you're all here. It's very nice to see a full hall for this 3MT regional competition. So I'm Josephine Nalbantoglou. I'm the Dean of Graduate and Postdoctoral Studies at McGill University. And I'm really honored to be chairing this, to be hosting. This is the first time that McGill is hosting the Eastern Regional 3-Minute Thesis competition. It's always a highlight of our spring to have our own 3MT, and we just had it about a month ago, so it's really wonderful to have it again a month later. And I'm very excited to hear all the speakers from all over Eastern Canada. I'd like to welcome them. I know how much work went into preparing for this, so we're all excited to hear you. I also hope that you enjoy your time in Montreal and that you've also had time to talk to each other and to network. Before we begin, I'd like to acknowledge that McGill University is located on land which has long served as a site of meeting and exchange amongst Indigenous peoples, including the Haudenosaunee and the Anishinaabe nations. McGill honors, recognizes, and respects these nations as the traditional stewards of the lands and waters on which we meet today. So I'd like a few notes for the audience. You can read them here. The first one is turn off your cell phones, please. We don't really want any distraction for our presenters because everything is really honed beautifully, and we don't want their attention taken away from that. You can take photos, that's allowed, but no flash, no sound. And we also have a People's Choice Award today. So you should all have a ballot on hand for this. And you will also have a chance to submit these at the end of the judges' deliberation. And for all of you who are watching us on live stream, we will be providing you with a link to vote online at the end of the presentation. So I think most of you are aware, 3MT is really a very popular competition. It helps everyone, not just the participants, but also the ones who train them and the ones who watch the presentation, hone their communication skills. And this has been developed at the University of Queensland in Australia in 2008, and now it's an international competition. It's really held all over the world. In Canada, the English competition is sponsored by the Canadian Association of Graduate Studies, CAGS. So today's presenters were all selected at their host universities, and they're representing their host universities for the privilege, really, to advance to the CAGS finals, which will be held later on. So in the competition, the graduate students have 3 minutes to present a compelling oral presentation of their thesis and significance to a non-specialist audience. You might all be specialists in your own fields, but you're not going to be a specialist in each field. So the participants know that they have to communicate the big ideas and their vision and their passion to all of you. So, and it's a single slide. So that is really the challenge of it, is to be able to project everything that they want to say in that one slide. So we have all our presenters, Peter Rosowski from INRS, Olivier Gazil from Polytechnique Montréal, Ogboko Kalu from University of New Brunswick, Laura Hall from Dalhousie, Milan Valier from Concordia, Georgia Darmonkov from Memorial University of Newfoundland, Matthew Dunlop from University of Prince Edward Island, Molly LeBlanc from St. Mary's University, Jean Lagacé from Université de Montréal, Jay Olson from McGill University, and Mohamed Suleiman from École de Technologie Supérieure. So welcome all, and we're really looking forward to hearing you. I'd like to also present our judges. They have a really onerous job of deciding who is going to stand out. And our judges are Lynn Slawaska. If you could just get up so people could see you. Mr. Christos Siros. And Chantal Srivastava. So I'll just give you a little bit of background on our judges. Lynn Slawaska possesses 25 years experience as an executive leader, manager, mentor, and coach in the areas of customer service, marketing, business consulting, and human resource management. She's a psychology graduate from McGill University and an MBA graduate from Concordia. She also holds the Certified Human Resources Professional designation. She's recognized for her passion for organizational excellence and transformation. In 2014, she was certified as a master practitioner in neurolinguistic programming. And 2016, she obtained the Proceed Change Management certification. A lifelong learner, Lynn lives in Montreal, and she's currently working as a general manager at Le Belmont. Thank you for participating. Mr. Christos Syros is a graduate of McGill. He's a former politician and cabinet minister in the Quebec government from 1981 to 2004. In 2004, he was appointed as the Quebec government's envoy to Belgium, and he was a key architect of the Canada-European Union Free Trade Agreement. From 2014 to 2017, he served as the Quebec envoy to London in the UK. He currently works as the director of the UK-Canada Foundation. This creates academic and non-academic partnerships in both countries. Thank you again for coming. And Chantal Srivastava comes from Radio-Canada. After graduating from Laval University's Public Communications program, she was approached by Radio-Canada in Quebec City, where she was responsible for the press review and public affairs. She has worked on VST Bonjour, Tournée d'Amérique, Les Actualités, Samedi, et rien d'autre. In June 1997, Chantal Srivastava won the Grand Prix of Radio Journalism of the Community of French Language Public Radio, CRFLP, for Kigali, Après l'horreur, la vie. It's a powerful report on the role of women in the reconstruction of Rwanda after genocide. So thank you all for coming and for participating in this, and I hope you find it as fascinating as we always do. So we have prizes. There's a first prize, and the winner goes to the CAGS final, as I said, but there's also a monetary amount attached to it, so it's $1,000. Second prize is $500. Third prize is $250, and it's the same prize for the People's Award. So all of you will have a chance to vote for your favorite presenter, as well as with the People's Choice Award. So the judging criteria, it's based on communication. So clear communication methods, so it's a comprehensive and understandable summary of the thesis, and the ability of the presenters to engage the audience and really teach them about their subject matter. That's what's important. After the last presenter, when the judges leave the room to deliberate, we will have a short question and answer period. So we will not be interrupting in between the presenters, but we'll only have one question period at the very end. So to the audience, you may also want to keep these judging criteria in mind. How they communicate, how well you understand what they say, and how they really transmit the impact of their work and how they engage the audience. So those are really all the important topics you should keep in mind. So I'd like to introduce our first presenter from INRS. It's Peter Rostocky. The title of his thesis is Generation and Control of Complex Optical Quantum States Using Photonic Silicon Chip and Fiber Technology. So this is a mouthful, and this is where we always tell people, give us a lay title. His lay title is Quantum Computing in Rainbows. Peter?
Quantum physics is weird, but it's just a description of how matter and energy interact at the single particle level. Quantum physics can also solve problems that would take centuries using typical computing. But I'm here to talk about the Mona Lisa. Current computers encode information as bits, a 0 or a 1, black or white. But if we encode information in quantum bits, we can store a 0 and a 1 in a particle at the same time, holding more information. The problem is that currently access to qubits requires very complex equipment. And at the same time, even per particle, a qubit is often not enough. This has meant that quantum computing has largely not made the transition from laboratories and science fiction. What I want to tell you about today is how our work, making use of optical qubits, can enable practical quantum systems and at the same time even more information. So we make use of light at the smallest scale. It's particles called photons to store our zeros and ones. And this concept of a particle being two modes at the same time can be counterintuitive. But if you think of yourself, you are currently occupying an x and a y coordinate in space at the same time. And this is exactly the same mathematics as for a particle. The difference being is that it can occupy an x and a different x simultaneously. This is known as quantum superposition. Now, the x's that we can make use of for photons can, for example, be its position in space. But since we're working with light, what we chose to use was its color and encode the 0 and 1 as different colors of a photon. And there's no reason for us to just stick to a qubit of 0 and 1. We can make an optical qubit, a photon that is simultaneously red and green and yellow and blue. You get the idea. We went up to 10 colors, having that much more information in just a single particle. We're also able to do this using a very practical means, generating the photons using just a silicon chip. But in order to manipulate the colors, to do information processing, do we require complex equipment? No. We borrowed standard equipment from telecommunications. So these are people who mix colors of light and fiber optics for a living in order to process and perform operations on the photons. So in summary, we created a system that you can take home and have an astounding amount of information in just a single photon. Our next work will focus on bringing this technology and pushing it towards even more meaningful quantum computing problems. But I think that we can agree. Quantum physics is still weird, but it looks better in color. Thank you.
So we learned something during our own 3MT competition, and that we went through very fast from one presenter to the other, and we didn't give the judges enough time to really think about what they want to do. So I'm here to kill some time before the next person comes in. And as the judges tell me that they're ready to proceed, I will introduce the next speaker. So we're going to have a little bit of dead time. Because they're really marking all the criteria. You could also probably exercise the same thing about it. Yeah, almost seems done. So I'll introduce the next speaker, who's from Polytechnique Montréal, and it's Olivier Gazil. He's studying chemical engineering, master's degree. He's had the boldness to do the competition with his colleagues of the PhD. So we've got a master's candidate here. He knows how to distance himself from his speaker's talents, and he has really the honor of representing the Polytechnique today. The title of his talk is Jello and Green Chemistry, a Golden Opportunity. We could ask him at the very end what his big, long title is when we're killing time in between. Olivier?
So what is the common element between this bride, Jack Sparrow, and this chemical reduction of an alcohol? Well, you would have guessed it. It is gold. This bride has a magnificent golden ring. Jack Sparrow is searching for gold, and this chemical reaction can be done with gold. But what is so magical about gold? Gold can accelerate chemical reaction, and they can do so by reducing the chemical usage. So good for the environment. But you must wonder how gold can do such a thing, since gold is a precious metal that does not corrode, that does not react. Well, interestingly enough, when gold is at the level of the nanoparticles, so really small, it becomes highly reactive and becomes an excellent catalyst. But there is a problem though. Imagine mixing some sand in water. Eventually, the sand will settle down. Well, for nanoparticles, it's the opposite. The nanoparticles stay in suspension, making them really hard to separate. So you need some way to immobilize them to be able to reuse these nanoparticles in different reactions. So current techniques to immobilize these particles are costly, they're complex, and not really effective. My solution? Do some jello. So imagine some jello shots, but replace the vodka with gold nanoparticles. So you put in the jello powder, the gold nanoparticles, the water, mix everything together, and voila, you have immobilized your gold nanoparticles. So currently I'm looking if lime flavor is better than cherry. Jokes aside, there is yet another problem. So the nanoparticles at the middle of the jello, well, they're actually out of reach from the chemicals. My solution to overcome this problem, so I use this special mold to be able to create all these interconnected tunnels, as we can see here on this picture, in the gel. So it becomes like an ant colony. This little ant or chemical can enter the tunnel, reach the nanoparticles, react, and then get out. So in summary, I have immobilized gold nanoparticles in a jello. So if you take the jello out, you take all the nanoparticles with it. Then I have create all these interconnected pores by using a special mold to be able to reach all the nanoparticles to optimize the reaction. And why use gold nanoparticles? Well, it's to reduce the use of energy and the use of chemicals. So it works toward green chemistry. So each time you have this tasty treat, well, think about green chemistry and how it can help the environment. Thanks.
Thank you very much, Olivier. Our judges are very concentrated. I think it's almost unfair that they have to work so hard and we're just sitting back and enjoying ourselves listening to these great talks. But I guess that's what comes with choosing or accepting to be a judge. So our next speaker is Ogbogu Kalu. He's a master's candidate in engineering at the University of New Brunswick, and he's tackling a very critical health problem that plagues nearly 1 billion people nationwide. And this is the problem of safe drinking water. And he's using a novel and cheap filtration combination of silica-based mesosporous materials and natural sunlight. Ogbogu?
We are facing a global water crisis, and the stakes are high. The stakes are our lives. According to the World Health Organization, about 1 in 6 people in the world currently lack access to clean water. It has been estimated that over the next 2 decades, the average supply of water per person is likely to drop by 1/3, possibly leading to millions of preventable deaths. Now, existing research for the most part has tried to solve this problem by using methods aimed at removing contaminants from water. And while some of these methods have resulted in contaminant removal, they tend to fall short in at least one of 3 key areas: cost effectiveness, sustainability, and the quality of clean water produced, meaning they aren't ideal solutions for our global water crisis. My research suggests that the key to our global water crisis lies in a simple chemical method that allows for the creation of a powder-like material whose particles look like tiny balls with nano-sized pores on them. How small is a nano-sized pore? So small that the thickness of one sheet of paper is approximately 100,000 nanometers. Now, you might look at this and you look at these balls and you wonder what are they. So these balls are made of silica, a versatile material that allows them to attract a variety of toxic waste, including harmful chemicals and bacteria to their surface. They're also coated with titanium, a material that allows for the sustainability of the process because it lets them use UV rays from the sun or any other human-made sources to break down the waste. So essentially, you have a nice multi-mechanism tag team combo, right? So going on, you find that if you look at water, what's essentially happening is it's attracting it, breaking it down over and over because the photodegradation allows for that sustainability. So because of sustainability, it also allows for the cost effectiveness of the process because sustainable solution, like a reusable solution, allows for a cost-effective solution. Now, aside from that, what's even more interesting is the fact that this method is done under like room temperature, unlike other methods that need high temperatures to operate, saving even more money. And aside from that, what's even more interesting is the different deployment methods of this material. You could use it to make porous filters for your water bottle or to coat a water tank the size of your dream home. Essentially, we have a process that's easy, versatile, sustainable, and cost-effective, making it an ideal solution to our global water crisis. Thank you so much.
Thank you very much. So we have two presentations on nanoparticles. There might be a message here. Is everyone actually taking notes on these, the way the judges are? Yeah? No? Yes? Maybe? So I'd like to present our next speaker, who's Laura Hall, a PhD candidate in the neuroscience program at Dalhousie. She previously completed her BSc at University of Western Ontario and her master's at Carleton. Currently, she's uncovering the secrets of eco-immunology, a field which combines her love of immunology with biology, ecology, and evolution. She's got an intriguing title, The Not Very Hungry Caterpillar. Laura?
Feed a cold, starve a fever is a saying that dates back to the medieval times. We now consider it to be an old wives' tale because why on earth would starving yourself while you have a fever help you recover from illness faster? The fact is that immune systems are huge energy sinks, and so it only stands to reason when they are fully operational, we should be providing our bodies with enough energy through eating to keep them operating on full swing. But, as I'm sure many of you will have experienced, when we get the flu, our appetite is the first thing we lose. This is called a sickness behavior, and it is a response generated within ourselves and not by the pathogen that's infecting us. Something really amazing about sickness behaviors is the fact that they occur in almost all animals. You feel lethargic when you get sick. So does your dog Lucky, your parrot Crackers, and even the ants that are marching up your driveway. That's right, everything from bugs to humans exhibit the same internally generated sickness behaviors. My research focuses on the fact that something so highly conserved must serve an important function. I use a caterpillar called the tobacco hornworm to study the enigma of illness-induced anorexia, or loss of appetite while sick. By measuring gene transcription, the presence of certain proteins, as well as behavioral assays, I was able to show why, in these caterpillars, the cessation of feeding during illness makes the difference between life and death. The immune system, albeit big, is not the only resource-demanding system within our bodies. One that is often overlooked is the detoxification system. Food is absolutely covered in all kinds of microbes, toxins, and other little nasties. The detoxification system is our first line of defense against these things, and it stands sentinel to ensure they don't become an internal problem. It turns out that there is a competition for resources between these two systems, and my findings suggest there are certain molecules, such as glutathione, which are vital to both systems. By allowing my caterpillars to abstain from eating while they are ill, they are able to shift all of these limited molecules towards immunity, and they can decrease their exposure to toxins by simply avoiding their food. If I attempt to activate both systems at once, there is not enough of these limited molecules to go around, and the caterpillar cannot mount an efficient immune or detoxification response, which results in death. So, although it may seem logical to want to increase our energy intake when we are ill in order to replenish the energy we are expending, there is more at stake when we are sick than just energy in equals energy out. So at least in this case, starving a fever is not an old wives' tale after all.
Thank you, Laura. I think the same thing happened during our 3MT competition, that we all felt we were so knowledgeable about so many different things as we walked out of that room. We forgot it very soon afterwards, but just the fact that we're in here listening makes a huge difference. The judges are writing all their comments. You know, we give them at least about a half an hour at the end to be able to just sort all of it out. So our next presenter is Milan Valier. He's a Concordia public scholar and a PhD candidate in the Center for Studies in Behavioral Neurobiology at Concordia University. He investigates the brain processes underlying addiction and has a passion for communicating science to a general audience. And his title is What Makes Us Want to Drink? Milan?
Take a second to think about your friends, your family, including a few generations back. Most of you can probably picture someone that drank a little too much, a little too often. Almost half of alcoholics trying to abstain from drinking relapse within 3 months. And historically, this relapse was thought to be motivated by withdrawal symptoms, where a person would resume drinking to ease the anxiety, tremors, and general malaise of sobriety. But what about relapses that occur after withdrawal symptoms have subsided? And many do. Well, if you ask someone who's trying to abstain from drinking or someone who's relapsed, what makes you want to drink, or what made you resume drinking? They will list cues, and usually 2 different types. One type are called discrete cues. These are things like the sight, smell, or taste of a preferred alcoholic beverage, the pop of a champagne bottle, or the kss of a beer can opening. Another type are called contextual cues. These are things like a night out, being at a party, or the ambiance of your favorite bar. These discrete and contextual alcohol cues that could once be encountered unproblematically at an earlier point in the addict's life can become the difference between sobriety and relapse. So the question I asked myself in the lab is, what are the brain processes that control how people will respond to these discrete and contextual alcohol cues? I work with a rat model of alcohol seeking because rats have very similar brains to humans. But in the rat, unlike the human, I can use cutting-edge neuroscience tools to shut down specific pathways of neurons. I can do this at a time when the rat is presented with alcohol cues and observe how they respond. Do they scurry over to a dish where they used to consume alcohol or not? What I've found is that alcohol seeking to discrete and contextual cues seem to be controlled by separate neural pathways. So when I shut down one of these pathways, I reduce alcohol seeking, but only to the corresponding cue. These fundamental studies will help identify targets for pharmaceutical intervention in humans, and that's the ultimate goal. If we can understand the neuroscience of what motivates a rat to pursue alcohol, then perhaps we can prevent a person, someone who really wants to stop drinking, maybe that person you imagined at the beginning of the talk, from relapsing. Thanks.
Thank you, Milan. Our next presenter is Georgia Darmonkov. She's completing her Master's of Science in Medicine, Clinical Epidemiology at Memorial University. Her research interests are in diabetes, specifically in the pediatric population. She hopes to help develop tools and protocols to reduce the number of preventable diabetes complications. And her title is a Diabetes Knowledge Translation Project. Georgia?
Good afternoon, everybody. So we are going to get personal right off the bat. So with a show of hands, in grade school, how many of you recall asking to go to the washroom during class? All right. And again, with a show of hands, do any of you recall ever asking to go to the washroom multiple times during a class? All right. So myself included, I'm sure we're all guilty of sticking our hand up just to go and roam the hallways. But did you know that asking to go to the washroom frequently and truly needing to go can be a telltale sign of diabetes? So unfortunately, diabetes has become a household word in my province. Newfoundland and Labrador has one of the highest incidences of diabetes in the world, with a child being diagnosed every week. So diabetes is a condition in which the body cannot produce or effectively use insulin. And insulin is the hormone which assists the entrance of glucose from your bloodstream into your cells to be used as energy. And our body battles with insulin deficiency by breaking down fatty acids. And when these fatty acids break down, ketones are produced, and they build up in toxic levels. And this acidic environment that is then created is called diabetic ketoacidosis, or DKA. So DKA is the leading cause of morbidity and mortality in children with diabetes. And the most important thing to note about DKA is that with proper insulin management, it can be totally avoided. So this is a very expensive preventable condition, as everyone with DKA ends up in the intensive care unit. So a number of years ago, the Newfoundland and Labrador DKA project was developed. This was a multi-phase knowledge translation project to better educate both healthcare professionals and the public about diabetes and DKA. So on the slide here to my right, you can see the project deliverables. So this project was implemented for a 2-year period, and my master's research focused on determining the efficacy of the project. So as part of my analysis, I looked at DKA hospitalization rates for the 2 years before, during, and after the project. And so what I found was that for the 2 years during the project, rates were significantly lowered, followed by a slight increase post-project. And I also determined that contrary to the current literature, the most hospitalized age group were the 20 to 24-year-olds. So moving forward, it will be beneficial to have this type of education campaign in place for a longer period of time to become truly ingrained in people's minds. And further, we must add a facet specifically targeting these 20 to 24-year-olds. And I think this could be achieved by individual education in the form of peer-to-peer interaction, as well as transition of care education for physicians. So in closing, this was a cost-effective method at reducing the burden of a preventable diabetes complication. So your take-home for today is to keep those bathroom visits monitored. Thank you.
Thank you, Georgia. Our next presenter is Matthew J. Dunlop. He's a PhD student working between the School of Sustainable Design Engineering and the Department of Chemistry at the University of Prince Edward Island. His current research seeks to aid local mussel fishermen by isolating high-quality nanocrystalline cellulose from locally invasive tunicate species. Matthew?
So tunicates are an invasive aquatic species which came to PEI waters a little bit over 20 years ago. And since tunicates showed up, they've been causing major problems for our local aquaculture community, and in particular for mussel fishermen. Tunicates release thousands of larvae every year into island waters, and these larvae are sticky. They stick to underwater surfaces, like mussel socks, which you can see in my slide. Now, the effect of this over time is that tunicates engulf the entire surface of a mussel sock, as you can see top left, your top right. Tunicates and mussels eat the same food, and the problem is that tunicates starve mussels when they surround their mussel socks. And the problem with this is, at the best, the mussels take longer to grow, and at worst, a mussel fisherman loses his entire yield for that year and kills every single one of his mussels. Current research is focused on mitigating and containing the tunicates that are in our waterways, but we want to approach that problem differently. To directly address the tunicate problem, we will harvest tunicates industrially, removing large quantities from the water and turning them into valuable products. Now, one of those products is cellulose-based. So nanocrystalline cellulose is simply a component of cellulose. You can break cellulose down into something smaller. You can think about it as a microscopic version of rebar. So cellulose we associate with plants. The floor you're standing on, that I'm standing on, has cellulose in it. Many other plant sources exist. But tunicates are the only animal source of cellulose that is known to science. And because it's synthesized in a tunicate differently than it is in a plant, there is a difference between the nanocrystalline cellulose you can isolate from tunicates versus what you can isolate from plants. If you think about nanocrystalline cellulose as microscopic rebar, then the rebar you can isolate from a plant is about 5 feet long. But what we can get from tunicates is much bigger, about 20 feet long. And if you think about using these in composite materials as a structural reinforcement, like a rebar is in a concrete building, then you can imagine that the applications for tunicate-sourced NCC, nanocrystalline cellulose, are much larger than plant-sourced nanocrystalline cellulose because the material itself is inherently more valuable. It is longer crystals, and we can't get those crystals anywhere else but tunicates. So this research seeks to industrialize the harvesting of tunicates in PEI waters to produce nanocrystalline cellulose of high value, introducing a new product to market which provides an economic incentive to harvest these tunicates in the first place. Now, simply put, it is very difficult to contain and mitigate nature. Our history tells us this in everything we try. But what we've been doing for thousands of years is simply being to harvest nature's bounty. We see tunicates as a problem, as a nuisance species, because they're causing us problems. But are they really a nuisance? Are they in fact a resource? Could they be used to produce something valuable, a new industry on Prince Edward Island, and solving the problems that are already associated with our mussel industry because of tunicates? That's the question our research endeavors to address and answer. Thank you.
Thank you, Matthew. So we've had nanoparticles, and I think another message that's coming through is sustainability and sustainability science, and what our graduate students are doing to address these problems. The next presenter is Molly LeBlanc. She's completing her Master of Applied Science at Mary's University in the Department of Environmental Science. She lives in Halifax, Nova Scotia, and is here to talk about her work studying the environmental effects of abandoned gold mines. Molly?
Gold, a metal that makes us think about wealth, value, treasure. Across countries and cultures, gold is revered, searched for with intensity, used for our most beautiful and important possessions. And in my province, Nova Scotia, we have a history rich with gold. We're no California, but we've been mining it for about a century and have produced over 1 million ounces. But today, those gold companies are almost all retired. And if you were to visit one of the 300 abandoned mines they left across the province, you would see something like this. Because prosper and development isn't all the mining industry left behind. In order to separate the gold from the rock, miners used to use another metal, mercury. But we know now mercury is a neurotoxin. It can accumulate in the kidney, the brain, even in a developing fetus, and cause serious neurological and developmental damage. For every 1 ounce of gold mined, 1 ounce of mercury was used, meaning over 1 million ounces total. And as you can probably guess, environmental protocols weren't as strict back then, so any leftover waste was typically just dumped nearby, usually in a wetland like this one. Today, there's thought to be 3 million tons of this sand-like, mercury-rich mining waste left in wetlands across the province, in Nova Scotia alone. But there are sites like this all across the country. Despite that, though, hardly anyone is researching the effect these sites have on the species living nearby. So that's where my work comes in. And I'm starting with the little critters low on the food chain, wetland insects. What I found is that insects from these gold mine sites have mercury levels 50 times higher than insects from clean, healthy wetlands. And in species like dragonflies and mayflies, which shed their skin, hatch from the water, and fly away, those adults are still keeping almost half of that mercury even after they hatch. So this means not only are these sites leading to mercury entering the food webs, it's also at risk of spreading, moving with these insects as they leave the wetlands and fly to neighboring habitats. Now, I can probably guess your answer if I were to offer you a choice between a handful of gold and a handful of bugs, but let me make my case for why these guys are just as valuable. These insects are the base of so many important food webs, some of them which lead to us. And the fishing industry is huge here. If our fish are feeding on insects that are high in mercury, there's a chance mercury could make its way to humans. And these sites are still open to the public. They're used for ATV rallies, for cottages, and this picture shows children playing in what looks like sand, but is actually tailings. Now, the price of gold is rising again, and we as a society put so much value on things like gold. But what deserves just as much of our attention and our passion is cleaning up these messes we make so we can protect the real treasure. These species and ecosystems we all depend on. Thank you.
Thank you, Molly. I think it's quite remarkable that we have a good mixture of doctoral students and master's students. So thank you for participating, even very early on in your graduate education. Our next presenter is Jean Lagacé. He's from the University of Montreal. He's from the Department of Mathematics and Statistics. And his thesis is making links between classical number theory problems and the equations governing the physics of waves and vibrations. Jean?
Hi. When I was a kid, whenever I'd go in the kitchen or the bathroom, I'd look at the tiles on the floor and wonder, what can we say about all those pretty patterns? How could I continue them under the bath, say, where I can't see them right now? Well, if you told me that 20 years later I would do this as a job, I would have told you, ha, that's not a job. Yet, here we are. An important question you can ask about those tiles is, how many of them do you need? That's a very important question if you want to tile a bathroom. Well, any tiler could tell you that the amount you need is approximately the area of the bathroom divided by the area of each of those individual tiles. Now, those tiles are a one-shot deal. You don't want to have any left at the end. You don't want to miss any. So you add a number of them enough to cover the perimeter. And the reason you need to do this is because the shape of the tiles don't fit perfectly inside the shape of the bathroom. Now, if you're in the bottom left corner case, this is not a problem, because the area is much bigger than the perimeter, so your first estimation was still pretty good. However, in the top right corner case, the perimeter and the area are of comparable size, so was your first estimation using the area still good? Well, my field, asymptotic analysis, could also be called estimation science. As such, we have all the right tools to answer these types of questions. Now, you can remark that, say, in any of those two cases, if you have one of the tiles that's around the edge, you will cut it to make it fit. And maybe the part that you cut, you could use it around another part of the edge. And as such, your estimation using the parameter would be a gross overestimation. Maybe you need less than what you added there. Well, one field that understands how to deal with those type of cancellations is in the physics of waves and vibrations. Here, when you have a wave that's comprised of many smaller waves of different frequencies, you might have constructive or destructive interference. And this destructive interference would be exactly this, cutting on one side and putting on the other side. A very important theorem in harmonic analysis tells us that actually counting those tiles is exactly the same thing as calculating the amplitude of a wave whose frequencies would correspond to the corner of those tiles. And that's exactly what happens. In my PhD work, I have shown that in the top right corner case, we have destructive interference. The area estimation was a good one. We don't need to use the parameter too much. Well, now you might tell me, a PhD in counting bathroom tiles, is that really serious? I mean, do we have more important things to do? Well, you'd be right, and that's the beauty of mathematics. The same model that I'm talking to you about today using bathroom tiles is the same model that tells you how many energy levels are available in a crystal, or that tells you how whiskey slushes inside a glass. Well, that's it. Even for the physicist, for the tiler, for the whiskey aficionado, the mathematics on which I'm working explains parts of their everyday life. Thank you.
Thank you, Jean. I think this could be a good outreach project, you know, how math is going to explain everything in everyday life. And you bring someone like Jean in from grade 1 and every year visit those classrooms to tell the children the same thing. Our next presenter is Jay Olson from McGill. Jay's got an unusual background. At the age of 5, a magician pulled a coin out of Jay's ear. So he became fascinated. And 2 years later, he started performing magic himself. So this eventually led Jay to study the psychology behind magic. And he now uses his knowledge as a magician to enhance healing and placebo effects. So his PhD is in psychiatry, and Jay adds a little bit of magic to modern medicine. Jay.
Matthias, 7 years old, was our first participant. He had suffered from migraines 2 to 3 days a week for years. But after an hour of our procedure, he stopped having migraines for 6 months. This is the power of placebos. Usually when we think of placebos, we think of a sugar pill that has no active ingredient but can still have profound effects. But placebos can be any kind of inactive treatment, like an injection with no active ingredient, or even a machine that does nothing but you still believe that it can help you. Now, placebos are the most studied procedure worldwide because they're often used as the control group, but they're one of the least well understood. We do know some factors that make placebos stronger. So complicated or expensive-looking procedures work better than simpler ones. So machines work better than injections, which work better than sugar pills. Children experience more effects than adults do. In my research, I combine all these different factors in order to use placebos not as a control, but as a treatment. Right now, we're focusing on 2 disorders, migraines and attention deficit disorder, characterized by fidgeting and reduced self-control. Neither of these disorders has an ideal treatment. They often require expensive drugs that can have serious side effects, even changing your personality. So we take children with these disorders and we introduce them to our novel treatment, a placebo machine. It's actually a broken brain scanner that looks and sounds like a functioning one. Now we tell the children, although this machine is completely inactive, it can help your brain heal itself through the power of placebos. In fact, placebos work even if you know that they're placebos. So we put children inside the scanner for about 15 minutes. We give them positive suggestions that this procedure will help reduce their symptoms and improve their strengths. A month later, we follow up with the families. 90% of them report improvements in their kids, from reduced fidgeting to better sleep. And in some cases, children report massive, lasting improvements, like Mateus, who, a year and a half after his initial session, is only having 10% of the migraines he used to. Now, these results are still at their early stages, but we're hoping to develop new placebo-based treatments that are safe, effective, and have no side effects. In doing so, we can harness the power of placebos and, more generally, harness the power of the mind. Thank you.
So we're actually already at our last presenter, who's Mohammed Slimane. He's a PhD candidate in power electronics at the École de Technologie Supérieure. He has a master's degree in renewable energy, and he's passionate about finding new, smart, and efficient technologies to harness green energy for electric grids. Mohammed?
Imagine it's January, and it starts to rain outside, but it's freezing rain. Then ice starts to accumulate on rooftops, on cars, on trees, and especially on electric wires and poles. Then the ice starts to bring down transmission lines, one after another. You find yourself without electrical power, unable to heat your house, unable to use the oven, and outside, metro stations are closed. Most of the schools, banks, and grocery stores are closed too. Well, that happened during the Great Ice Storm that hit southern Quebec in 1998. The problem was, without an alternative transmission line, there was no way to bring back electric power anytime soon. People were very generous with their neighbors, as they were sharing their blankets, their food, among many other things. Now, if this were to happen again, would we be able to avoid such a massive outage? Good afternoon. My name is Mohammed Slimane, and my research is about designing methods that ensure flexible and secure supply of electrical power without interruption. For this, I study modular multi-level converters, or MMCs. Now, MMCs are powerful devices used to convert electrical energy resources with the electric grid. And as compared to traditional power converters, MMCs are more modular, flexible, and efficient by design. Very similar to what engineers or kids can do with Lego blocks. What's most important is that if one component fails within the MMC, you can still use a neighbor component, and this makes our whole system fail-proof. Now, the challenge is MMCs are complex in structure and difficult to operate. That's why in my PhD thesis, I've chosen to design simple yet powerful statistical methods to operate the MMC. So now we are able to reduce the operation complexity of MMCs without compromising their performance. Now, for instance, one can use an MMC to charge an electric car at night. And during the day, one can recharge the batteries and use the excess energy back to the grid. Now, today you are dependent on electrical power more than ever before. At the same time, we are committed to reducing CO2 emissions. And with the help of MMCs, we can use and share in a smart way a variety of renewable energy resources to support our grid. Now, if another great ice storm happens again, like in 1998, thanks to the MMC, this time Your neighbor could offer you more than just blankets and wood to keep you warm. They could offer you their energy too. Thank you.
As I said, Mohammed was our last presenter. So now we're going to have the judges' deliberation. But first of all, I'd like to have a hand for all of our presenters. All done a great job. So I'm going to ask the judges to leave the room with all their notes, their binders, their pens. So that they can negotiate the prizes. That's fine. That's fine. So we're going to open the voting for the People's Choice Award. So for the people who who are here on site, you have your ballot on hand. So you just check off one box next to the name of the presenter that you're voting for. For those watching online, you can vote using the link that's here on the screen, or by clicking the link in the description of the video on the YouTube. So take the time to vote, because People's Choice Award is an important award also. And we're going to let our presenters get up, mingle, take a drink of water, and so on. And then they'll come back to answer questions from the audience. 5 minutes.
Everybody, you can hand your People's Choice ballots to Bara here. Lynn. So you have to collect your ballot.
this year it's usually.
Perspective of we're moving forward.
I'd like your attention for a few minutes. It's your 2-minute warning for the end of the vote, for people's choice. Get in your choices in there. Every vote matters. Thank you.
It was really good. And they got good. So this is the second year that they did it that way, and it makes a huge difference.
Now good to see how other people are participating at this level, and you may not be the top, but you can see who's sure.
Yeah, yeah, yeah. And that's what we're saying, that like ours had like a bit more.
So we're almost there. Just before we announce the winners, I'd really like to thank the judges. I never volunteered to be a judge because I think it's such a difficult job. So we have a few gifts for the judges. Olivier and Sophie, I think. Olivier's here, so he'll do it. This is our thank you for you.
Thank you.
Thank you very much.
Thank you. Merci.
And again, you know, it's too bad that we have to have winners and others. I think everyone's a winner today. So I want so a big hand to everyone who participated. You know, it was terrific. We all loved it. We didn't get to have a question and answer period, but we're going to have reception afterwards, so everyone will be picking your brains, actually. So get ready for that. So we're ready? Yeah. We're ready. Okay. So we're going to do the certificates, and we're going to start with the People's Choice Award, the first one. And the People's Choice Award goes to Molly. Thank you so much. Amazing. Okay, so I think we just get a...
Wow, thank you. Awesome, thanks.
Congratulations.
Oh, can I? Yeah.
Okay. Third place goes to Molly. And actually, I made a mistake initially when I was announcing that only the winner went to the CAGS final. It's the top 3 who go to the CAGS final. So great. The Eastern Regional sent 3 candidates to the CAGS final.
Amazing.
Congratulations.
Thank you. Awesome. Yay. Yeah. Oh. Thank you so much. Thank you.
Congratulations. Second place goes to Jay Olson. Congratulations, Jay. She's tweeting. And we have this ridiculously big check for the winner, $1,000. And the winner is Laura Hall. So we're going to give you... So you put this into your luggage.
Okay. Thank you.
We also have this.
I'll take that. I'll trade.
Yeah.
Thank you.
So now it's our reception, right? Okay. So it's been our pleasure. This is the first time we've hosted the Eastern Regionals. I'm willing to host them every year, actually. It's so much fun. It's worth having it here. So we just like double up the pleasure that we have with our own 3MT and then the regional 3MT. So congratulations to everyone. I'd like to thank the audience. You've been very, very good. You've been quiet during the live streaming. No phones, no flashes, nothing. And I hope you enjoyed it as much as I did. I'm sure you all learned amazing stuff. And we have all the participants and the judges remaining for a few minutes afterwards because we have a reception. So join me in congratulating everyone again. And I thank you for participating. So we're going to have actually part of what we have to do before the reception.